All right. Hello, students of SBI3U and whoever else might be watching. This is the first attempt at any kind of video lecture. So I've been spending the day just kind of playing around with my settings and getting set up. There's no way this isn't going to be awkward, but I'm still going to post it. Hopefully the idea is that I'm going to get used to these video lectures, just like you guys are getting used to these video lectures, as we, carry on through this self-isolation period. Let's talk about learning goals for today. I've kind of gone ahead and set these up. So here's our learning goals for today. Number one, we just want to get used to the video lectures. That's the first step. Me getting used to making them, you guys getting used to watching them. Number two, we're going to just review the sexual reproduction note. If you recall, the last note that we saw in class was this one on sexual reproduction. So that's what we're going to do. We're going to review that note for the advantages and disadvantages, and some strategies. We watched that video about the slugs mating. That kind of thing. Let's go get those learning goals out of there. I can bring them up anytime I want. Another thing that I've decided would be helpful is to have the current topic always there at the bottom of the screen. So here at the bottom of the screen, there at the bottom of the screen is going to be the current topic. I'm going to try to keep that up on screen for most of these lectures. That way when you're jumping around the YouTube video, you can just be like, oh yeah, this is the topic I want to be on. Oh, that's the wrong topic, things like that. Right. Let's see. What else? So that's that. Let's just jump right into getting used to video lectures. Why video lectures? So here's the other thing that we're going to have is I have a thing that I can do on top, which is, I call it keep in mind, but I'll have a thing on top that I can say, hey, here's why I'm rambling on about whatever I'm rambling on about. So let's start with talking about why I'm making this video. Well, number one, you can pause and rewind YouTube. Let me just fix that to rewind. Yeah, pause and rewind YouTube, you can do that. You might note that a lot of your teachers are just using Google Hangouts for lectures or whatever they might be doing. This way I can have you guys get your content early and then kind of figure that out later. Number two is attendance is easier. Attendance is easier for me because I can just put quizzes online, and when you guys do the quizzes, that tells me that you've seen the lecture, and we're good to go. Number three, the ministry. The ministry wants proof. The ministry wants proof that you guys are learning. The ministry wants proof that I have done my due diligence as a teacher. So the videos being online and publicly accessible and the quiz results tell me that I can show that to the ministry, and then you guys can get your credit, which is great. So let's go ahead and clear all those out, and we'll go keep in mind. Keep in mind that you guys need 10 hours a week. Wow, look at that. I'm already doing bad typing. Keep in mind, you need 10 hours a week of content. So 10 hours a week of course content is our guideline. We can't give you a credit unless you've got, I think, 90 hours for the entire class. So what this is going to work for us is your 10 hours are 5 hours on these lectures. Ooh, still messing up my typing. Here we go. 5 hours on these lectures and 5 hours of live help sessions. Now, I know in my email and my PDF, I said that I would be doing those live help sessions on Google Hangouts. I've actually changed that. It's going to be on Keep. I had a look at Google Hangouts and I had a look at Keep, and I really like the features of Keep more. Like, you guys are able to like scribble on the notes and things like that. And also I can mute you if I don't like what you're saying. So I prefer that to the Google Hangouts version of this thing. Those 5 hours of live help sessions, you guys have to log in once a day, right? You have to join the meeting at least once a day. You have to contribute in some way. You must contribute at least once per session in some way. So asking a question, highlighting something on the note, solving a practice problem, whatever we're going to do there. And the last piece is it's a total of 5 hours per week. So yeah, total of 5 hours per week. So you could do 2 hours one day, 2 hours another day, and then 1 hour, and then just kind of pop in and contribute something when we're doing other stuff. That way you guys can choose where to send your attention. If there's a particular topic that you have a bigger, longer question about, you can spend more time there. And if it's like, oh yeah, I got this, then just kind of get your 5 hours in, get a couple of minutes in, contribute a little bit, and then peace out and wait for a longer time. Please don't just do token stuff at the start of the week and then get in 2 hours on Thursday and Friday. I feel like that would not be great. Okay, so that's that. Let's pull up this keep in mind. Keep in mind, we are going to go ahead and say this video is not mandatory. This video and its quiz. This video and its quiz are not mandatory for your 5 hours or 10 hours. This is just for you guys to test out the thing. How are we doing so far? We're 8 minutes in. Okay, great. So let's go back over our learning goals. Our learning goals are getting used to the video lectures. I'm feeling better already. Hopefully you guys are too. So I think we can strike that one out and we can move on to actually going over this note. I think what's going to happen, I think what I've decided to do is, it'll be maybe a 45-minute video, 15 minutes for the quiz. So we're going to try to do something like that. We're going to try to do a 45-minute video and about 15 minutes for the quiz. We're done getting used to video lectures. And I guess I was going to show off my cameras. Let's show off the cameras that I've got for you guys. So this is the front view. You've got my desktop view. You've got my desktop view with me up in the corner here. You've got my desktop view with me over on the left. Oop, the calculator is active there. Oops, sorry, let me just turn off the calculator. Okay, great. Me in the left. You've got the webcam over here. Hey, there's a webcam over here. Hello. That's actually the one that's getting the audio for us right now. And you might notice this shiny surface right here. Well, that's a whiteboard that I stole from Miss G's math class. Thanks, Miss G. And that is here. This view is the document camera. So, if I go over here and I grab a marker, you can see me. You can see me writing. That's me. That's the camera. There's a camera right up here, and it means that I can write stuff. I can draw something. I could maybe draw a fake little coronavirus here. This is kind of what this virus looks like. These little things are proteins. It uses that to sneak into your cells and then drop its DNA. Well, RNA actually. It's an RNA virus. It's RNA load. We can draw stuff and we can erase stuff. So that's going to be useful, especially during those help sessions. So yeah, that's what I've got planned for you guys. I'll go back to the front camera, and here we go. Okay, we're 10 minutes in. Let's change our current topic to review of meiosis. Meiosis. There we go. So current topic is now going to be review of meiosis. Keep in mind, we're going to try to keep this video to 45 minutes. I'm going to have a little test on it, a little quiz on it later to get your 1 hour. I hope videos will be about 45 minutes long. Sometimes they'll be longer, sometimes they'll be shorter, depending on what we're doing. But yeah, let's go over meiosis. Okay, so let's review some stuff. First off, review meiosis. Actually, you know what? Not meiosis. Mitosis. Let's review mitosis first. We're going to review mitosis first. I found this really cool video. Of, let me get my face up in here. There we go. I found this cool video of mitosis, actual animal mitosis, a real time lapse, and I'm not sure quite how long it takes. There's actually a timestamp up in the corner here, but you guys might not be able to see it. Let's watch the video, and you'll see just how long each stage takes. And then we'll go through the video step by step so that we can identify each phase. So here we go. I'm going to mute my camera while the video is playing because otherwise you'll get reverb. Okay, here we go. Okay, turns out that video was actually silent, so I didn't have to mute my webcam at all. So sorry for that awkwardness, but like I said, it was going to be an awkward thing no matter what we did. So let's watch this video again, and what I want you to do this time as you watch it is keep in mind how long did each stage take. So keep in mind, I'll change this over to a question. Question, how long did each stage seem to take? How long did each stage seem to take? Or better yet, what was the longest stage? And if you don't remember what the stages are, let's review that. It's prophase, metaphase, anaphase, telophase, cytokinesis. So that's our question, is how long did each stage seem to take? Alright, so we're going to play the video again. We will try to identify these stages. Here we go. Right. Let's figure out how long these things took. We might actually just jump over to the document camera, and I think I've actually got it where I can put the screen up too. Yes, I can. Okay, let's actually use our timing down here and figure out how long each stage took. Um, our stages are prophase, metaphase, anaphase, and telophase. Telophase. And I guess cytokinesis. We'll just put these seconds of time lapse that it took, to get through these stages. So obviously we're starting at 0 seconds. Let's play the video. And the difference between prophase and metaphase is in metaphase they have to line up in the middle. So let's have a look. When we're looking up in the corner there, they're condensing. They're condensing. They're kind of doing a wiggle. If you looked at this under a microscope, would you say that that's metaphase, or would you say that that's still prophase? I'd say it's probably still prophase. I don't see a really good lineup happening yet. But right here, I'd say they're starting to really get that lineup going there. And that is at 27 seconds. So getting all that spaghetti, all of those chromosomes kind of lined up together takes a while. Now, from here under the microscope, I'd probably say it's metaphase. Let's keep going. Okay, that's definitely like there. If I saw it in a microscope, I would definitely call that anaphase. So 27 seconds to 34 seconds on my video is metaphase. So clearly it doesn't take nearly as long once they've kind of decided to line up, once they've kind of shuffled around and organized each other. All right, now anaphase itself starts at 34 seconds, and you can imagine it's not going to take very long because all it has to do is go out. Maybe out to there. Like that's where they kind of lose their little kind of feel. So that happened at 39 seconds. And then telophase. So telophase is when they're actually going to go back into chromatin, back into spaghetti instead of chromosomes. Cytokinesis is just going to be the membrane pinching in. So let's see how long that takes. You can already see the membrane pinching in there. And they're done, right? There's just this little tiny bridge there. But they're already dissolving. They're dissolving back into their chromatin pieces. And that. So that happens at 45 seconds, 39 to 0, 45. If you saw that under a microscope, if you saw that under a microscope, you'd probably say those are two separate cells by now. I'll finish looking at it, and yeah, that's it. So, I guess cytokinesis is it happens during that time. Looking at the times then, the longest phase is probably prophase or prophase metaphase. It's kind of hard to tell when one starts and one finishes. Some people, some biologists actually put them together into something called prometaphase. It's kind of like you saw them shuffling around. So it's like, are they just condensing or are they also getting into metaphase? We don't really know. So, so depending on which biologist you ask, there's like either 3, 4, or 5 steps of mitosis, depending on whether they call telophase and cytokinesis the same stage or prophase metaphase the same stage. Everyone agrees on anaphase because it's got that really obvious I'm getting pulled away thing from under a microscope, but the other ones are kind of hard to tell, especially telophase and cytokinesis. They happen at the same time. Right, so there's a review of mitosis. Meiosis is going to look very similar. I think I've actually got a video of that, so let me see if I can't find it. I do have a video of meiosis. Just as a review of meiosis, the goal of meiosis is to, well, first off, let's change our thing. Meiosis. Current topic, review of meiosis now. And the question is, why are there more stages? Right, the current question is going to be, why are there more stages in meiosis? I'll jump back over to our to this view here, where the question is, why are there more stages in meiosis? And we'll watch this 1 minute video. It'll hopefully help explain it. This time I will actually mute my my camera, because this one does have sound, and you'll get to hear the person talking. I'll put on the captions too. Okay, here we go. I'm going to mute my camera.
Meiosis is a special type of cell division necessary for sexual reproduction. These are sperm-producing cells of a male crane fly, a species with only 8 chromosomes per cell. During meiosis, the cell divides twice, producing 4 haploid cells containing 1 copy of each chromosome. The first meiosis division separates pairs of homologous chromosomes, creating 2 haploid cells with only 1 set of chromosomes. The second division separates the chromatids in each chromosome. The end result is a production of 4 haploid cells, each with a single chromatid from each chromosome. When haploid sperm and egg unite during fertilization, the resulting cell is diploid, having received 1 chromosome of each pair from each parent.
And now I'm unmuted again. Yeah, so that's meiosis. You can see a bunch of different meiosis stages happening here. This one's clearly in the middle of a cytokinesis kind of thing. There's some kind of hard to tell. If we move on through though, you got your usual prophase, then they line up for metaphase. They get pulled apart for anaphase. This is showing you the number they have. So yeah, these fruit flies have normally 8. That's their diploid number. As soon as they split once, it's haploid, and then the gametes are actually even less chromosomes than that. But we'll go over that as we jump into the note that I've got for you all. So close that. This we'll get to later, the interactive meiosis, but for now, let's do our review. Let's just jump back to our learning goals real quick. We haven't talked about the advantages and disadvantages of sexual reproduction yet, so we'll have to get to that in a second. Maybe we'll actually just flip those two. No, I'm not going to flip those two. Okay, so let's talk about this stuff. The keep in mind now, we're going to change over to a keep in mind. We are interested in the number of chromosomes at any given point. So here's the keep in mind. We're interested in the number of chromosomes at any given point. Okay, so here we go. Let's get started with that. So what is sexual reproduction? Obviously reproduction using 2 organisms. They have these things called gametes. That's just the general term for a sperm or an egg or like a pollen or whatever. Plants have pollen, whereas animals have sperm. Both of them still have eggs, which is kind of interesting. Gametes have half the amount of genetic material that was mentioned in that video. If it wasn't half, then you increase the number of chromosomes with every generation, and that's just, untenable. When 2 haploid gametes fuse, they make a diploid zygote. That was mentioned in the video as well. So you get 1 set from mom and 1 set from dad. All right, offspring are different from parents. This is in line with our learning goal. Just as a reminder, our second learning goal is to review the advantages and disadvantages of sexual reproduction. So this part here says, this can be a pro or a con. The pro, of course, is you can get a genetic advantage because, if you get, a gene, two different genes for example, muscle mass from mom and from dad, you get more muscle mass than either your mom or your dad. You can have, a child that's more advantageous, something that's more fit to their environment, as we learned during the during the evolution unit. Sorry. So that's that one. Actually, I should change the current topic, shouldn't I? Current topic is the advantages of sexual reproduction. So yeah, the advantages is, as we learned in evolution, in that evolution unit, that variation is good. So keep in mind then is that variation is a good thing for a population. Why is variation a good thing for a population? Well, it means that natural selection can act on that varied population. It means that the fittest will survive, will pass on those good genes. So sexual reproduction allows those good genes to proliferate a lot faster than asexual reproduction. Now the downside, of course, is you have variation the other way. It's a curve. So I'll just jump over to the document camera for a sec here, and we'll just erase all this stuff. It's a curve. So variation is good, but it means, if these guys live, if these guys are fit, these guys are unfit, having extra variation means you have more chance of this happening, but it also means that you have an equal chance of, these guys happening, the really unfit. Now, of course, we're talking about reproductively unfit. So yeah, with sexual reproduction, you get that nicer curve, that bigger, wider curve. If you're talking about asexual reproduction, basically your genetic diversity looks like this. Right? That's basically what your genetic diversity looks like with asexual reproduction, because you're making clones of the same thing. That's great if the fit line is over here, but if something happens in the environment and the fit line moves over to here, you're kind of screwed. Pardon the pun. Okay. Moving back to our desktop view, our standard view. So you can have genetic illnesses, you can have those recessive traits like we talked about for recessive traits. Here's the bubble boy, David Vedder. He had severe combined immunodeficiency disorder. I think that's what SCID stands for. He had to live his entire life in this plastic bubble. Eventually they expanded it to a whole sterile room. And then he died pretty young. I forget how young he died. So yeah, his parents were okay because they had one good gene, one bad gene, one good allele, one bad allele of that gene. But he got both of the bad alleles, and yeah. But variation is good. So right here, variation is good, and that's what you'll see up here. Variation is a good thing for population, kind of invariably good. The bigger your population, the more likely you are to have individuals that are going to survive. Okay, moving on. Let's talk about gametes. This is the current topic then is, gametes and sex cells. Right, gametes and sex cells. Gametes are your sperm and your ova. They differ from organism to organism. Typically, we say that typically the difference is that the sperm can move and the ova has to get pushed kind of through a uterus kind of tract. The ova is usually bigger. It has like mitochondria and starter proteins and things like that, so that the cell can, the new zygote can have everything it needs. I like to call it the starter pack sometimes. Right. Most sexual reproduction is between a distinct male and a distinct female. When we don't get into like hermaphrodites and things like that. And the male is defined as the thing that the organism that produces the sperm, and the female is the organism that produces the egg. Typically, the female will carry the children as well, with some exceptions. Like seahorses. Seahorses, the males will carry the kids. The females still produce the eggs, but then they transfer that fertilized egg into the male, and the male is the one that carries the kids. In seahorses, that's why we say the male carries it. The female creates the eggs, but that doesn't necessarily mean that she's the one that gives birth. It's kind of an interesting example for seahorses. Then you got the hermaphrodites. So those have both male and female things. So let's go ahead and ask a question here. Question. Why would hermaphrodism have evolved in certain populations? Right. Let's ask that. Why do we have certain things like certain types of bugs and mollusks and things like that are actually hermaphrodites? I think it's mostly mollusks. So take a minute, think about that. I guess you could just pause the video for a minute and think about it. The advantage, of course, is that when two organisms meet, automatically they make a breeding pair because they've got both kinds of organs, right? And not only that, when you have a male and a female thing, only one of them gets pregnant, only one of them carries the kids, only one of them actually, does the reproducing part. When you have hermaphrodites, they can both fertilize each other. So like earthworms and leopard slugs and stuff, they will fertilize each other. So both of them get pregnant. You get twice the offspring in one mating kind of event, which is useful for them. So that's that. That's gametes and sex cells. That's our question. Moving on. Gamete production. Right. I guess we don't really have a thing. So let's go back to a keep in mind. Females are defined by making eggs biologically, rather. Keep in mind, biologically, as the individual that makes ova. Right, females are defined biologically. Females are defined as the individual that makes ova. Right. So in mammals specifically, we have testes that produce the sperm, ovaries produce the ova. The sex cells are made by this meiosis process, which is what we saw the video on and what we're going to go over. We have about 10 minutes left, so let's jump right into it. Here's the steps of meiosis. Current topic is the steps of meiosis. So current topic is the steps of meiosis. Let's change over to the keep in mind. Keep in mind, meiosis wants to take a diploid cell and make 4 haploid gametes. Here's the keep in mind, right? Meiosis wants to take a diploid cell. Our goal is to take a diploid cell and make 4 of these haploid gametes. Here's the steps of meiosis: prophase, metaphase, anaphase, telophase. We skip prophase, metaphase, anaphase, telophase, cytokinesis. So, prophase looks the same. Nothing special there. You pack up your stuff. It stops being spaghetti, so it can move a bit better. You start getting the centrosomes moving. You get the spindle fibers starting to kind of attach. One weird thing that happens here is the crossing over. So, crossing over. One from mom, one from dad, you have 23 pairs, making your 46 chromosomes. They pair up and they'll sometimes intertwine. Here's a great picture of it. So the picture of it is that they'll cross over, and so not only do you get half from mom, half from dad, sometimes you get the sperm will actually have 60% of dad's chromosome, 21, and the other 40% is mom's chromosome 21. So you get even more variation, and just as a reminder, variation is good. Back to our view. So, metaphase, same thing. They align along the equator. As we saw earlier, this actually takes a while. The prophase-metaphase line is really, really hard to see. Anaphase, they get pulled apart. That's under the microscope, it's really obvious. They look like they're going, oh no, I'm getting pulled apart. You end up with 23 chromosomes. Each chromosome is a set of chromatids. In humans specifically, 23 chromosomes, right? Different things, different animals, different plants and everything, different numbers of chromosomes. So each cell suddenly becomes haploid. Right? Chromosomes reach the opposite poles, they aligned. Right? All that happens. And you end up with the daughter nuclei, which should say haploid rather than diploid. Sorry, that's awkward. Let me fix that. Haploid, daughter nuclei. Yeah, haploid daughter nuclei. So here's the overview. I start with 2N. 2N is the number of chromosomes, right? Humans have 23, so our typical body cell actually has 46 chromosomes because, you get 23 from mom, 23 from dad, and all your cells have those 23 from mom and 23 from dad. It splits to N and N. Right? So that's that. Now we skip over prophase 2. There's no prophase 2. We go straight to metaphase 2 because these things are still packed up. So it's easier to go with. It's easier. It's already packed up, so it's easy to pull apart. Anaphase 2 then happens. The same thing happens. It gets pulled apart. And there's telophase 2. Same thing that it looks like. You got 4 haploid daughter cells. So we use haploid for both, and that's kind of an awkward thing. So I'm going to try to explain that to you guys back over on the document camera. We're going to erase our fit and unfit, and we're going to change what our current topic, diploid versus haploid. All right, current topic, and keep in mind the diploid and haploid is based on the number of chromosomes, not the number of chromatids. So we'll put that up here, right? We'll put that up there. Chromosomes, not chromatids. What do I mean by that? Well, when you look at a chromosome, what you'll often see, or what you'll see drawn a lot of the time, is something like this. Something like that. and those are chromosomes. This is sometimes called a chromosome. Right, but each side has a chromatid. So one chromosome actually kind of has two chromatids in your body. Again, one from mom and one from dad. So normally you've got 46 of those, and in meiosis 1, what you'll see in the first stage of meiosis, is that at the start you have, you know, your chromatids floating around because they've been duplicated. Then they'll line up in the middle. But they're paired up, because what's happening here is they've actually been duplicated. So they're paired up. Then when they split like this. They split, but they're still duplicated. Right, and then you end up with two cells. Right, each one with half the number. So, right, you end up with two cells, each one with half the number. So if the normal number, if this number is 4, right, this is 4, this is 4, this becomes 2. Right, that's the chromosome, and that's the chromatid up here, right? So there's 4 chromosomes, right? If we look up here, only the amount of chromosomes, not chromatids. You know, I'm going to take away the laptop screen. Let me do that. Right, chromosomes, not chromatids. So yeah, chromatids, chromosomes, still 4, but down here we're down to 2 and 2. Alright, so 2 chromosomes, not 2 chromatids. There's still, you know, there's still 4 chromatids here. If we kind of have the chromatids, there's 8 actual individual chromatids here, and there's 4 here. So we've cut everything in half. Yeah, we've cut everything in half. I'll just try to move that a bit more. There we go. 4 chromatids. Yeah, and then in the second part of meiosis, meiosis 1, so this is meiosis 1, this is meiosis 2, those 4 chromatids in 2 chromosomes, right? It's 2 chromosomes, but 4 chromatids. 2 chromosomes, but 4 chromatids. Those are going to turn into, you know, they get pulled apart actually at the chromatid level instead of the chromosome level. So you'll end up with still the same chromosome number. Right, still the same chromosome number because it's still those 2. Right, but it's actually just 2 chromatids. Just 2 chromatids. So that's why we say that this is all diploid. So this is still haploid, even though we've actually done. So this is haploid. This is still considered haploid. Still considered haploid. Yeah. So that's telophase 2. Back to this, right? Each cell necessarily has different DNA. Yeah. So yeah, this is a confusing diagram because it says N to N, and you're like, but it didn't duplicate. It didn't do that. Well, that's because chromosomes are not chromatids. It's still the same number of chromosomes, it's just a different number of chromatids, and that's an important thing to think about. I'm going to add that to the quiz for this thing. Yeah, we're just about running out of time, but here's a good example of this. Right here, if it's N, here N would be 2 chromosomes. You still got the same 2 chromosomes, or you still have one purple and one red, so N is still 2, but the number of chromatids has gone down in each cell. This has 4 chromatids on 2 chromosomes. This has 2 chromosomes, but only 2 chromatids. It's important to talk about. Everything's independent. You know, you can figure out just the chance of you being born out of your specific mom and dad's, you know, crossing over. It's insanely small. It's, you know, 1 in 7 trillion, 1 in 70 trillion. It's something ridiculous. Last thing is gametogenesis. So gametogenesis is our current topic. Back over here. We make the gametes. The sex chromosomes are X and Y specifically for humans. This is different for other animals. So for us, females are XX and males are XY, but for other animals, sometimes you know, it's like WW and WZ, or like AB and BB, and other ones actually have like multiples, not just 2. There's all kinds of weird things that happen there. Yeah, and those are karyotypes, and you can figure out the karyotypes. There's a specific stain. It makes specific bands in the on the chromosomes. We can identify the chromosomes by their bands. So for example, this is a male and this is a female. But if you look at the bands, let's find one that's kind of obvious. How about chromosome 16? He's like there's like 3 bands that are really obvious there. You see the same 3 very obvious bands on this chromosome 16. So it's like it's you get hints as to which chromosome you're looking at just at the banding pattern. These days, of course, computers can do it for us. What's wrong here? You have XXY. That's a Klinefelter's male. What's wrong here? You've got trisomy 21. That's Down syndrome. Yeah, of course, computers can actually do karyotypes. So you can just have a computer do the job for you instead of having to, you know, carefully pick it apart. People used to actually do this by hand. You'd have to actually pull the chromosomes apart and get them all on a piece of paper, well, not a paper, but like a piece of plastic, and then photograph them. Yeah, and that's the end of the presentation. So hopefully that worked out okay. I hope you were able to follow the video without too much trouble. Let's review our learning goals real quick. Hopefully you're more used to these video lectures. I feel a little bit better now. We're going to review the advantages. We reviewed the advantages and disadvantages of sexual reproduction, particularly when it comes to genetic variation, and some reproduction strategies like hermaphroditism, gametogenesis, things like that. The advantages, the specific advantages of hermaphroditism. Right. So please check in on SBI and try the quiz that's associated with this video. I'm off to go make that now. Thanks for paying attention. At least I hope you paid attention, and I hope I'll get better at these, and I hope you'll get, you know, more confident with these as we move forward. Just as a random aside at the end of the video, the fish are okay. So I brought the fish home. There they are. Yeah, so the fish are fine. Okay, that's my first isolation video done. So here I am signing off. Thanks, guys.